And this is not only something that coming from my laboratory, but it's also an organization that we set up to work on rare genetic disorders. It's called World Without Gene Biopsy. This is also part of a presentation from there. Basically, I'm going to talk about neuromuscular disorders, and particularly genetic neuromuscular disorders. And these are essentially, just to give you a brief background, affects peripheral nerves and muscles, because nerve effect would also lead to muscle defects. Ability to perform voluntary movement, most of it, like you want to raise your leg or you want to raise your hand, ability to walk, all this gets affected. Disability quite often leading to complete paralysis, in the sense that people can be completely ambulatory, wheelchair bound within a very short time from the diagnosis of the disease. Almost all these diseases are progressive. Okay, so it's not just a kind of disability you see that in an accident, or you see in many cases. It changes, it slowly becomes worse. And there can be genetic and non-genetic origin. Roughly about 80% of them are genetic origin. So there are a large number of causes of neuromuscular disorders. I will not go into it. We will be concentrating on the ones that are caused by genetics, mutations. You do see, because of variety of other infections, to the diabetics also actually eventually causes some of the neuromuscular problems. So how many of them are there? There are more than 1,000 such diseases that affect, very large number of them. The thing is that some form of this therapy is available for roughly about 1% of these diseases, okay, but not cure. Hardly any cure is available for any of these disorders. Though for those diseases where some form of therapy is there, the therapy can be as expensive. For some of the lysosomal storage disorders that affect muscles, the therapy can be as much as 70 to 80 lakhs per patient per year, just to keep them alive. It's not a cure. Finding cure is the only choice we have. So our organization is essentially directed towards finding cure for these diseases. And there's hardly any effort is going on in the whole country, and very little effort going on all over the world. So how many of such, I said more than 1,000 such diseases are there. There are a whole lot of different classes of these diseases, okay? And if you look at the individual number of patients of each disease, it may not be that much. Some cases like Duchenne muscular dystrophy, you have quite a few, but some others could be like 5 patients or 10 patients. But if you look at collectively, then the numbers runs into crores. Okay, so it's a major health problem, public health problem in the country, and in many cases the symptoms are very similar. So if you look at collectively, it is a major problem, but some others we don't see this problem, and no effort is being made on it. The other thing, if you look at it, these are the different classes of neuromuscular disorders. The number of genes or loci that are involved, and if you really total is 519 genes are thought to be involved in different neuromuscular diseases. So you can see that is a fairly large fraction of the human genome, or total number of human genes that affect these diseases. They are not small number that we are talking about. So what is the complexity of these diseases, and why there is a major problem? Because there is no link between genotype and phenotype for most of these diseases. We don't have any understanding of it. In many cases, only diagnosis has been made, or it has been shown that the mutation in particular gene causes the disease, but we just don't know how that thing happens. Okay, unlike, for example, red blood cells, some of the diseases of it, thalassemia, etc., you know very clearly that this is the molecule, this is the disease that happens. I'll just give you some examples of the complexity that happens. For example, multiple genes can cause a similar phenotype. For example, spastic paraplegia, and number of genes that are involved here, and as you can see, that very large number. I've only listed some of the genes that mutation in these genes would actually cause this symptom of that. Okay, so the gene and these genes all have a variety of functions. Cytochrome P450 to others, DNA repair enzyme, etc., etc. So there is essentially no link, essentially you see, between the kind of phenotype you see or the genotype you see. Again, multiple genes can cause the same disease. There we saw that again multiple genes can cause this. This is a very specific gene disease called limb-girdle muscular dystrophy. This is much more specific than the previous one, the spastic one that I said. And you can see that a whole lot of different genes, again, they cause it, the similar disease. The most of these have been diagnosed, very clearly understood, but we don't know how this relationship between genotype and phenotype is. Then the complexity is much more because you see that different mutation in the same gene can cause multiple different diseases. Okay, for example, this is ryanodine receptor gene RYR1, and all these different types of diseases are caused in the mutation in this particular gene. So you have multiple genes causing similar disease, and you have different mutation in the same gene causing different disease. So, and that increases the complexity of the problem. And in terms of this genotype phenotype problem, this is really a severe problem. And for example, if you want to relate target genes with, let's say, muscle function, so you have a muscle degeneration and you want to know whether, you would expect that the genes are known to be involved in a muscle specific function should be involved in it, but that's not true. I've listed some of the genes, target genes, which are known to have some kind of muscle specific function, like myosins, alpha-actinin, which is involved in this thing, and these are known. But on the other hand, there's a whole large number of genes which have nothing to do muscle. They should affect every cell type. For example, this is the one causes Pompe's disease. It's involved in lysosomal storage function, and lysosomes are needed by almost every cell. Why muscle? Why defect in this gene causes muscle? We don't know. And so are this, for example, GNE gene, which is involved in sialic acid biosynthetic pathway, and why it affects only skeletal muscles, not even smooth muscle. So these are some of the problems that are associated in that. Some examples of mutational complexity that comes in this, different kinds of mutational complexity comes in different kinds of disease. You could get something like a polyglutamine extension, that is expansion of polyglutamine, and that is seen in X-linked spinal or bulbar muscular atrophy. So you can see that. You will see duplication or deletion in the regions in Charcot-Marie-Tooth syndrome, okay, or you see a non-coding triplet expansion, okay. So non-coding triplet expansion that you see in myotonic dystrophy. You also see deletions and things happening in spinal muscular atrophy disease, and interestingly, there are large number of de novo mutations that occur, and these are the mutations that is not inherited, that occurs later on, and this also being found in many cases. De novo mutations are a major problem because if diseases are occurring because of de novo mutation, your prenatal diagnosis will miss it out completely, and that's the major problem. Large number of Duchenne muscular dystrophy you see because of de novo mutation that's happening. So severely recessive or early onset peripheral neuropathy, you see de novo mutation, you see majority mutation, the skeletal muscle alpha-acting gene is because of de novo mutation, and you see in both Nemelin myopathy and Lane distal myopathy, you see a lot of de novo mutation. So they are, and consequence of de novo mutation, as I said, is that there is an absence of family history, and which will not allow the whole, for example, screening mechanism based on prenatal diagnosis will not work. It will not work at all, and also leads to somatic mosaicism where your body have different cells. Some cells could be normal, and some cells could have mutant allele that is coming. So it really creates problem in terms of your whole management of diseases, early diagnosis, etc. So what is the reason for lack of therapy? I said the major problem is that if you are a patient of it, there is nothing else you can do other than waiting for things to become worse, because there is no cure for this, no therapy for majority of these things. So why there are? So one thing is that, of course, scientists don't want to work on problems like this. Everybody, if you talk to any students, wants to solve problem of cancer or diabetes. So they are not thinking about this. So because the number of patients are very few in a specific disease, that there are not too much of interest either among researchers, dot makers, or granting agencies. The major problem is that we don't understand the biology of these diseases. And if we understand the biology of the disease, I think it will be much easier for us to figure it out. Figure it out a drug for them. Of course, lack of resources and research reagents. For example, you are trying to find a mouse model which mimics the disease, they are not there. You will find for cancer, you find for diabetes, but you can't find for this. Absence of clinics then handle this patient is another problem. For example, a large number of neuromuscular diseases, particularly of adult type, the ones that occur among children, quite often the pediatrics people are quite familiar with it, but the adult myopathies or dystrophies, there are essentially 2 or 3 centers in the whole country where some kind of diagnosis, other things can be done. Current regulatory processes are not compatible with ground realities, but I must say that the new clinical trial guidelines that has come up, for which our organization actually made a major plea to change the rules, because the rules regarding the number of patients that you should have in the groups and this should have, that should have, really doesn't go along with the current situation of it. If you want to put minimum patient as 50 or 100, finding 50 or 100 patients for trial can be very, very difficult for some of the rare disorders. So some changes have been made. In fact, current regulatory guidelines for clinical trials has one section for rare disorders where a lot of rules can be overcome by that, and that's because some of us actually put forward our ideas regarding this. So I just take 2 examples of a reasonably well-known disease versus not, just to give you an idea about how much biology and other things we know. Duchenne muscular dystrophy is a very well-known pediatric neuromuscular disease. Roughly about 5 to 7 lakh patients that India has on that. And it affects birth in 1 in 3,500. This is quite common. There are more than 200 mutations that cause any of the forms of this disease. And it is a dystrophin gene which is affected, is the largest gene in the human body. And more than 100 exons that are there. It is the most severe form of the disease. Generally, the patients may live up to 30, 35. But this is also the disease where maximum amount of research which is going on. And the biology of it is reasonably simple. This is the dystrophy gene, as I said, very light. This is actually the one that connects with the external environment. It helps the signal from outside to go into the muscle cells. The actin is involved, and there are a number of proteins that are associated with it. In fact, mutation in any of these proteins would also lead to a dystrophy-like symptoms that is there. So the biology of this has been worked out very well. In fact, people know the protein quite a bit, and they also know that what we can do with the protein. For example, exon 51, there is a mutation that causes the translation machinery to stop there. So it produces a truncated protein. So the effort has gone on. In fact, there is a drug out in the market that allows skipping of that exon to happen. So if you skip it, you produce the rest of the protein and it doesn't stop anymore. But that exon 51 is not needed. So even they have now come out with, they call mini dystrophin gene, where they got rid of many of the exons and produce a smaller gene, because that can be then produced through a AAV vector as a gene therapy option. So we know a lot about it, partly because the number of patients in the world is much more, and Muscular Dystrophy Association in America and other countries have lots of money to support. Other diseases also occur because of mutation. Dystrophin gene, Baker muscular dystrophy, cardiomyopathy dilated, X-linked cardiomyopathy dilated 3B, etc. So the same gene mutation would cause a lot of other diseases, and as you can see that mostly muscle-related disease in it. The problem with Baker and many other diseases affect all muscles, including your diaphragm muscle or your heart muscle. So essentially people die because of the fact that either you stop breathing or your heart stops because of the failure of the muscle function. The other gene which is hardly anything is known is DNA myopathy, which is our organization very interested in. It's a recessive genetic disorder, affects only skeletal muscles, that also spares some called quadriceps. Late onset, 20 to 55 years, progressive loss of skeletal muscle function. It starts with distal muscles. Diagnosis is clinical examination, gait analysis, pathology, and DNA sequence. Roughly about 200 diagnosed patients that we have right now. So I want to present 200 diagnosed patients, and we did a little bit of study here looking at data all over the place. It's a recessive, mainly compound heterozygous, that means two separate mutations in two parts of the gene. A few homozygous, you see, as I said, that 200 documented cases of DNA myopathy, and our organization has a record of most of these patients, so we know their mutation, etc. So we carried out a study and found that 70% of the Indians carry one of the pathogenic alleles is common among 70% of the Indians. It's not seen anywhere else. It's only seen in Indian subcontinent or the Indians abroad who are migrated from India. So when we looked at this pathogenic allele which is present in 70% of the patients, using genome data wherever it is available, we did an analysis of that. We found that 1.5% of all Indians, normal Indians, carry that pathogenic allele. It's a heterozygous situation. And if you look at Gujaratis, this is community-wise, 15% of the Gujaratis carry this pathogenic allele. So and based on these frequencies, we calculated actually the estimated number of patients, which will be something like 30,000 to 50,000. The question is, where are the patients? Why we are seeing only 200 patients? And thanks to because of our organization website, we keep getting emails. And I just got an email 3 days back. And the email from a patient, he says it took 10 years for him to running around trying to get this diagnosis done. And still the diagnosis is not fully confirmed. And they wanted our organization help. I keep getting now emails almost every other week from somebody or other doing it. So what the diagnosis involves? A clinician must know and have seen this patient before. And most of the clinicians don't know anything about it. And I must thank this academy because this academy gave us funding to organize a workshop in Ahmedabad, and we took a whole bunch of neurologists and gave them some idea about how to diagnose and how to treat neuromuscular diseases. And I must thank that really worked out. Almost more than 50 clinicians participated and many students participated in the workshop. So other issues in diagnosis is an adult onset. Lot of people are married and lot of people are just pre-marriage state. The families we have talked to have really don't want to do it. They don't want to know what the problem is because it has a lot of social implication in it. So we believe that, we need to do a lot more surveys, identify this patient, get an idea about what is the true number of patients in this country. So DNA myopathy, I just said that we hardly know anything about it, mostly missense mutation, and null mutations don't exist. Though it's a celiac acid biosynthetic pathway, we don't know why only skeletal muscle gets affected. So, celiac acid biosynthetic pathway, hyperciliation seems to be a key molecule, maybe a possible mechanism. There is no good animal models exist, so we don't know. There are alternate mechanisms, and this seems to be more coming stronger day by day, that GNA gene actually participates in, it has a moonlighting function, does other things including cytoskeleton function. So, and that's something that we need to understand. We need to understand why cytoskeletal muscles are affected in this case. There can be multiple mechanisms that can happen, and this can be a tremendous biological problem for a basic scientist to take up. And all neuromuscular disease can be a great biological problem, and solution to this problem can really help us find cures for these diseases. And you can actually see a whole lot of different mechanisms that can possibly participate. I just point out one. If it is problems are because the mutation causes misfolding, then you have smaller molecules that can actually help to fold the proteins better. So you can actually come out with something called pharmaceutical chaperones as a possible therapy for it, provided you know that this is a misfolding problem. So genetic disorders provide natural system for basic human biology research, because you have, here I showed you that 513 genes are affected. Where will you find 513 genes? These are natural models for you to do gene function analysis, and I think this is these are like great problems that scientifically can be done and scientifically challenging. So I just want to point out in the end an alternate way to drug discovery. I mean, trying to propose this is that patient supported or patient participated drug discovery system. Patient groups are not considered to be stakeholders in drug discovery process. There was a recent discussion in DBT about new drug discovery. As I talked about clinicians, researchers, pharma companies, and they stop it there. And the patients are the most interested body in finding drugs, and we need to make them participate. They know what they want, and they have the highest motivation to get anything done, and we must take them into account. A tripartite collaboration, researchers, clinicians, and patient groups are needed for this purpose, and we have to make this system going in. There are, in America and Europe, there is a great now effort now going on to get patients involved in outcome measures. For example, if you are doing a drug trial, they're asking patients, are you feeling better? Rather than going through all the hundreds of clinical tests that increases the cost of running a clinical trial. Okay, or endpoint, what do you want? What should be the endpoint of a clinical trial? Patients are the best one to tell, this is what I want. And this is, after this trial, this is what I'm feeling. Their point should be coming. We have to think of an alternate paradigm in doing that. Patient feedback, etc. And patient can also support trials and partly research. Our organization is now sort of ready to start with funding small seed grants in strategic areas where we think is the fastest way to get to the drug. So I sort of thank a whole lot of people, and Sudha, my wife, she's been working in Dr. Nalini from Nimmans, Kadilkar. These are the two people who run clinics where you can diagnose neuromuscular diseases. They have the widest experience in it. Ashram Menon has been giving us a lot of sequence data for us to analyze, and that's been a great thing, and Partho has been a great help in doing that. Patient volunteers, families, And I must add trustees of our organization who have been a great support in getting this thing going.